So good afternoon, everybody. It's a real pleasure to be here in Southern California again. Thanks to the ARM for the opportunity to present Anocca and provide an update to you all. And thank you all for coming and watching the presentation this afternoon. So Anocca is an engineered TCR T-cell therapy company, a biotech based in Stockholm in Sweden. We have a mission to essentially decode the immune system to then recode next-generation T-cell therapy. And I'll explain a little bit more about what we mean by that as I go through the presentation. So just to introduce Anocca a little bit and kind of set the scene for what I'll talk about in the next few minutes. We are a fully integrated TCR-T company, meaning that we have R&D capabilities as well as our own in-house manufacturing capability. So over the last 10 years or so from the founding of the company, which incidentally was an idea on a piece of paper when we started, we've built what we think is a unique target and TCR discovery engine. That's generated an asset library, a TCR asset library of over 40 products to date. And as I mentioned before, we have then our own in-house TCR-T manufacturing capability, and we'll be actually taking our first programs into the clinic during 2024. Today, so we're a privately funded company. We've raised about $150 million from more or less the same dedicated group of private investors. We have around 100 employees in the company, which are mainly scientists based out in our research labs, in our manufacturing facility. Quite an extensive team of developers that are working with bioinformatics and other software tools that we've developed to support our platform. We've got a robust patent portfolio of 19 granted patents to date, and we're operating in around an 8,000 square meter facility in Stockholm, in Sweden, as I mentioned. So just to take a little bit of a step back and orient everybody, TCR-T essentially leverages the immune system. And in our case, and in the first indication areas that we're taking our technologies into, we see this, the opportunity in terms of targeting cancer. So as most of you will know, the adaptive immune system is one of the body's most precise defense mechanism. And at the heart of this are T-cell receptors, which precisely then direct the T-cell immune response. And what we're trying to do with TCR-T then is harness this ability to identify and kill diseased cells. And if only it was that easy. Essentially, there are three critical components that are required then for a T-cell to identify and eliminate a diseased cell. And taken together, this all provides a combined challenge for TCR-T developers. So if we look, first of all, at the TCRs themselves, so these are receptors, obviously on the surface of a T-cell, and the immune system is constantly producing huge numbers of unique T-cells with unique TCRs represented on those T-cells. The thymus produces around 10 to the 18 unique TCRs during development and during early stages of life up to early adulthood. That number's a quintillion, which is an enormous number, and that diversity is really required to prosecute then the antigen universe that the immune system is presented with during life. In terms of selecting the right optimal functional TCRs, what we need to ensure is then that we select the right TCRs from that enormous variety, and also that we select TCRs that lack off-target effects so that they can be used as treatments, as therapies in multiple individuals. So TCRs are constantly, or T-cells are constantly scanning, as I mentioned, this antigen universe. In terms of targets, so antigens are then presented to T cells. So these are infected cells, diseased cells that have become cancerous, for example. And the types of antigens that are presented on the cell surface are dependent on both the disease target and the HLA gene composition of an individual. And HLAs really drive patient segmentation in the context of TCR-T. Each person, in terms of class 1 HLAs, has 6 different genes, and each HLA can then present a different set of antigens. So you can see that this equation rapidly builds up to an incredible amount of complexity. And it's really unlocking this complexity then that we think, in terms of what we're doing at Anocca, that you require an assumption-free approach to find the right targets and then the right therapeutic TCRs. So what we do in terms of the platform that we've built is we bring together cell biology, modular technologies, and also software solutions then to replicate the immune system in the lab. And at the core of this are a series of precision functional assays that we've built. These are proprietary assays. And what these allow us to do in a very precise, controlled, and then scalable manner is actually analyze antigen presentation and also carry out TCR discovery. And this really is an assumption-free approach. So it's functional biology that is actually driving the decision-making within the prosecution of targets and TCRs in the platform. This is enabled by adding to this to these precision functional assays a series of scalable modular technologies. What this does is allow us to both standardize the performance of these functional assays and also integrate a level of automation into what we're doing. And this is really to allow us then to handle the enormous amount of data that we're generating and also operate the platform with speed and precision. All of this is underpinned by an in-house purpose-built software platform. This allows us to orchestrate then these complex workflows that we're working with on the one hand, and then on the other hand, we can automate then laboratory activities, everything from inventory through to handling of our robotic systems. And then we've also built a number of purpose-designed data analytical tools that again allow us to work with this high degree of complexity and data that we're producing through the platform. So just to show that in a slightly different way, the functional assay part of the platform is all built around programmable human cells that recapitulate then T cell immunity. So we have engineered antigen presenting cells, engineered TCR presenting cells. And in terms of the performance of the platform and the way that we've structured our capabilities, we're actually carrying out millions of analyses simultaneously to solve this complex HLA target TCR equation. And then the platform then operates at the speed and scale that is needed to crack this code. And I'll show you a little bit of data to show validation of the platform in a second. And clearly we can apply this then in several different disease areas, including oncology, infectious disease, and autoimmune disease. And the current focus in the company is in the oncology space. So in terms of the way that the platform performs and the type of data that we are producing, What we're seeing consistently now that we've established the technologies, we've integrated and built the platform, and we have that now functioning in the way that we envisaged originally. And what we can do is also generate then TCRs that are both potent and highly specific for a selected cancer target of interest. The graph on the left here just shows a very simple example of a peptide pulse assay. So this is from actually our lead KRAS G12V program. And here we have T cells expressing our KRAS G12V TCR, and we've pulsed this with a concentration range of peptides. And you can see it's classic EC50 concentration curve, and we're generating highly potent TCRs with an EC50 in this case of just under 40 picomoles. And if we look as well then in killing assays in a range of cancer cell lines that have different HLA densities, you can see in the graph on the right that we can consistently kill target cells even at low HLA density. And this is the typical type of data that we're producing within the platform. We have additional assays that we are also using, which are for analyzing cross-reactivity, so off-target activity of a specific selected TCR, and also alloreactivity, so off HLA activity of a specific TCR. And this allows us then in a preclinical setting to select TCRs that are essentially clinically actionable. So how do we deploy this type of approach then clinically? So as I mentioned, what we're generating are TCR-T libraries that then have broad population coverage and in terms of HLA depth and penetration, and then covering a diverse array of cancer targets. And we think this then offers a new treatment paradigm. So thinking about a monotherapy, where we can deploy an HLA-restricted TCR to a specific patient segment. And also, we are working with the approach of a multiplex-type approach, so deploying selected TCR-Ts to tailor an individual's tumor in terms of the targets that they're presenting and matching that to their HLA background. So this just shows then the pipeline. It's focused, as I mentioned, on oncology. We have a number of programs that are targeting shared driver mutations, other programs targeting shared tumor antigens, and then also additional programs targeting shared oncoviral antigens. And we'll be taking our first KRAS G12V programs into the clinic during next year, and they will be followed by other KRAS targeted TCRs behind that. In terms of manufacturing, as I said in the beginning, we have our own in-house GMP manufacturing facility, a 5,000 square meter footprint. We're supporting both autologous and allogeneic manufacturing. Our initial programs are in an autologous setting, as I just mentioned, and what we're then also gearing up to do, following on from these initial program developments, is to move to a more allogeneic manufacturing modality. So just to land then on this final slide, if anybody is interested in learning more, just reach out to me afterwards. I'd be happy to answer any questions, have any discussions. From a partnering point of view, we're very interested in partnering around our oncology pipeline, working with developers towards commercialization. We're looking at bringing in complementary manufacturing technologies, effector cell enhancement, different ways of delivering payloads, in particular looking at allogeneic platforms, so IPS-type platforms differentiating to T cells, and then also looking at therapeutic area expansion, so into infectious diseases or autoimmune diseases. So with 5 seconds left, I thank you for your attention. And as I said, please reach out to me afterwards and say hi and let's have a discussion. Thanks very much.